The organ that we all care about is this one, the brain, of course, which is enormously complex. There are almost 100 billion neurons in the adult brain, and together they make about 100 trillion connections. And the brain, of course, is a computer, a very complex one, and so what we're trying to understand is how that computer functions and how it's built up. And our understanding of the nervous system has been historically driven by technological developments. Ramon y Cajal is a father of neuroscience, and he was able to understand individual neuronal shapes and their connections because of the Golgi stain method, which allowed you to look at individual neurons. And then electrophysiologists were using recording methods to understand where the electrical impulses come from in the nervous system. So where are we now in terms of understanding the brain? I would say we're somewhere in the middle, probably closer to the beginning. We're asking the right questions, I think. We're making some headway, so to speak, but we are still very far from being able to say anything definitive about how the brain functions and how to fix it when it goes awry. So I'll be telling you about two new technologies that we're developing to better understand brain circuit function. The first is real-time visualization of synaptic strengthening, and the second is visualization of electrical activity. And I'll try to explain to you why I think this is analogous to an IKEA delivery truck and a intercontinental airplane. So when we learn, it's because new firing patterns emerge in the brain. For example, this region of the brain, the hippocampus, when you learn a new place or a new person, there are cells in your hippocampus that now fire more strongly than they did before. And that change in firing pattern is due to the growth of individual connections, the synapses, on the dendrites of those neurons. And it's been known for a long time that protein synthesis is required for stable synaptic growth and memory formation. And a hypothesis has been built up that proteins are synthesized from RNA molecules that are near the synapse in response to activity at the synapse. And those new proteins then help to build up the synapse, make it bigger and stronger. So we wanted to know, can we mark growing synapses by looking at new proteins, and how is this process affected in neurological disease? It's a complicated slide, but basically all you need to know is that we've developed this protein tag, which is a self-destructive fluorescent protein. So it emits light, except that it destroys itself at the very beginning. But if you want to see, you can put this on a protein that you're interested in. If you want to see new copies of that protein, you add a drug that inhibits this destruction. So then you add a drug, you can see where new proteins are going. And we believe this is useful for visualizing synaptic structural growth in networks in a way analogous to, let's say you wanted to see in a city like Boston, where the growing households. Well, growing households need new furniture. So one way you can visualize growing households would be to tag furniture as it leaves the IKEA store, and you can see where those tags end up. So this is how it functions. These are neurons on drugs, basically. We add this drug that allows us to see where new proteins are synthesized. In this case, we're looking at PSD95, which is a major synaptic structural component. And we think PSD95 is interesting because it's required for synaptic growth, and it's the main structural component. We want to be able to stimulate some synapses on the neuron, but not all. To do that, we plate neurons in these two compartment chambers. Two compartments are connected by tunnels that run through this barrier. And the tunnels are very thin, so only the dendrites, the processes on which the synapses reside, can project through the tunnels, and the cell bodies are stuck on one side. So we can now stimulate on one side with drugs that mimic synaptic growth, that induce synaptic growth. This one is BDNF, brain-derived neurotrophic factor, and it stimulates receptors that are required for the synapse to grow. And what we see, we ask, where do new copies of PSD95 go on the stimulation? We see that the synapses that are in the tunnels receiving the highest amounts of BDNF have preferential accumulation of new PSD95. So that does show that new PSD95 is a marker of growing synapses. We can see that as well with stimulation of a neurotransmitter receptor, mGlu-R. We see the same thing on the stimulated side, a lot of new PSD95, much more than on the unstimulated side. What's interesting is those experiments used a complete gene of PSC95. If we take out a region of the gene called the 3' untranslated region, we don't see that effect. So instead of seeing accumulation of PSC95 here, we see what just looks like gradual diffusion of new PSC95 from the cell body. It's a very different pattern of expression. And the 3' untranslated region is a part of the gene that's required for its transport, for the RNA transport into the dendrites, and it's known to bind Fragile X mental retardation protein. So we believe that the effect that we see is probably requiring FMRP, and we'd like to now look at neurons from disease models to see if what we see is different in a disease model. And we believe that new PSC95 will be useful as a marker of growing synapses. So we'll go to the second project, which is real-time visualization of electrical activity in the brain. So you can have a static map of the brain, like lights in a city, or you can have a dynamic map, which is more useful. And in this case, what you're seeing is airplane tracks as the day goes on. And you can see as dawn breaks on each portion of the world, the number of flights increases. And also, you can see where those flights are going. So you can see which cities are connected and where the activity is on the globe. So you can see how busy Europe is, for example, and where those Europeans are going. So in a sense, we want to do the same with the neurons in the brain. We want to see when they're firing and how they're linked together in time and space. People have been using calcium sensors to do this because calcium enters cells when they're firing action potentials. But calcium sensors are not perfect. They're slow to respond, whereas the actual voltage changes on the membrane take 2 milliseconds. Calcium takes 200 milliseconds to come in and out. And so also calcium doesn't see any subthreshold inhibitory activity, which is also very important. So to see this, we've been developing a sensor that directly monitors voltage in the neuronal membrane. We call this ASAP. It's called an allosteric sensor of action potentials. And you can see here an electrical trace and a fluorescence trace, a light trace with ASAP. And the two look very similar. So ASAP is following each rise time and each fall time. And even with subthreshold depolarizations, which calcium would be silent to, ASAP could see it. Whereas the previous sensor, arc light, you couldn't see it at all. It was, you just don't see the same pattern. With ASAP, we can follow very high trains of action potentials because it's a very fast responder. So you can, for example, have 200 hertz bursts of action potentials, and ASAP could follow each one, whereas arc light flattens out at 100. So we now have this sensor that we think is a very reliable sensor of membrane firing. It's very fast, it's very bright, and we like to use it to understand how neurons that are from patients with developmental disorders have abnormalities in synaptic function. And it's great to follow up on Dr. Urban's talk because we're also interested, and maybe we should talk about this, we're also interested in taking cells that are derived from patients, putting in our sensor, and then visualizing with high throughput in a very easy way how they're different in terms of their neuronal firing patterns. So in summary, I've told you about two new technologies for understanding brain circuitry. The first one is real-time visualization of synaptic strengthening, and the second one is real-time visualization of electrical activity. And I think both of these will be useful in providing some insight into how brain circuits function, how they're changed by activity, and how that might be affected in disease. So I should acknowledge Roger Chen, in whose lab I started the first part of the talk, the work in the first part of the talk, with help from a student and a technician and a collaborator in my own lab. I show the work done by Yang, Ying, and Francois, and these entities provided funding. Thank you.